Hello everyone. So here I was dealing with class 11 Ncert biology photosynthesis chapter. So we are in part 3. So in the previous lecture we have already dealt the non-cyclic photophosphorylation. I very properly explained there, so you can go and have a check. And here we'll be discussing the cyclic and non-cyclic phosphorylation. So before we go through the Ncert theory, it's important that we first understand the difference between these two. Okay, so when we come to non-cyclic photophosphorylation, you see in non-cyclic one, here both the photosystems are included. First here PS2, then PS1, and its product is going to be two products, ATP as well as NADPH. Both the products, these two are the products of the light reaction, as we know from the earlier lectures. Okay, and here non-cyclic, because this will not again, cyclic will be when this is getting repeated, but here it is not like that. It will go and it is not going to return back. Okay, that is why it is non-cyclic. But if we come to cyclic phosphorylation, here only one photosystem is included, that is photosystem 1. Here in non-cyclic, both the photosystem 2 as well as 1 both are included, but in cyclic only photosystem 1 is included. Right? This is the first difference. If we need to differentiate between non-cyclic and cyclic, first is in non-cyclic both the photosystems are included, but in cyclic phosphorylation only one photosystem is included. In non-cyclic photophosphorylation, the two products are ATP and NADPH, but in cyclic photophosphorylation only one product, that is ATP, is formed. NADPH is not formed here. Non-cyclic, here you see a Z scheme is noticed. You can say that a Z scheme is associated with non-cyclic phosphorylation, but there is no such Z scheme in cyclic phosphorylation. From light will fall here, PS1 electron will get excited. Okay, it will go to the electron acceptor, the electron will go to electron transport system, and it will return back to PS1 itself. Okay, started from PS1, it will go like this, having a cyclic, and it will again return back to PS1. That is why it is cyclic. There is no PS2 here. Here there is no PS2, we need to remember. Okay, rest for PS1 it is P700, we know it only. Here PS2 680 nanometer, PS1 700 nanometer. So this is the basic difference between the two. Here it is Z scheme, here it is cycle. Okay, so we'll go to the theory portion now. So in this portion there is nothing much written, so we go directly to the next page. In the earlier lectures I have mentioned that in the chloroplast there was mentioning of the ribosomes. Ribosome 70S was mentioned earlier. So chloroplast was associated with ribosome, means chloroplast do possess ribosome, but we also need to know that chloroplast is also associated with ATP. ATP is synthesized by the cells. Mitochondria we already know only, the powerhouse of the cell, and it's also by the chloroplast, and that is why it is named as phosphorylation. So here the definition of phosphorylation is given. This was supposed to be given earlier only, because they kept on mentioning phosphorylation in the previous pages, but they are telling the definition here. So ATP synthesized by cells is named ATP is synthesized by cells, and this process will be known as phosphorylation. Okay, production of ATP, formation of ATP, that is only phosphorylation in the presence of light. Okay, then it will be photophosphorylation. There is a difference between phosphorylation, means just production of ATP, but if the production of ATP is in the presence of light, it becomes photophosphorylation. So now the next is photophosphorylation is the synthesis of ATP from ADP and inorganic phosphate in the presence of light. This is something we know it from class 10 itself, right? Okay, so when the two photosystems work in a series, first PS2 and then PS1, a process is called as non-cyclic photophosphorylation, which I said this diagram. This is non-cyclic photophosphorylation. Z scheme, non-cyclic, same thing. Okay, so they are saying that your PS1, PS2, PS2, PS1, it goes on that way. The two photosystems are connected through an electron. The two photosystems are connected through an electron transport chain as seen earlier in the Z scheme. Both ATP and NADPH are synthesized by this kind of electron flow. As I already mentioned, in cyclic there is only one product, ATP. ATP-C. Cyclic phosphorylation is basically just to increase the efficiency of the photosynthesis. We need more energy, that is why cyclic will take place. But main is the non-cyclic only, because it is giving us the food, because it is giving us NADPH also, ATP also. That why non-cyclic is very important. Cyclic is just to increase the efficiency. It will give out some extra ATP which we need. Okay, so that was all about non-cyclic. Now in this paragraph there is mentioning about the cyclic phosphorylation. When only PS1 is functional, the electron is circulated within the photosystem and the phosphorylation occurs due to cyclic flow of electrons. So here the cyclic flow of electrons will take place from PS1. It will go this way and then it will come back to again PS1. Okay. A possible location where this could be happening is in the stroma lamella. Very important location. Location is also another big difference that I forgot to mention. But first of all, the product is a thing. The pattern in which the electrons flow, that is the second difference. And the third difference is the location. PS1, PS2 is included in non-cyclic. Non-cyclic will take place in the grana and the cyclic will take place in the stroma lamella. Now let me show you once again where the stroma lamella was and where the grana was. So here this is the grana. So here in this grana what will take place? Non-cyclic phosphorylation. Z scheme, everything will take place here in this grana. But two grana is connected by this joint, no? Stroma lamella, can you see? So in this stroma lamella, there only PS1 is going to be functional. There PS2 is not there in stroma lamella. PS2 lacks, stroma lamella, mark my words, stroma lamella lacks PS2 system. Okay, it lacks PS2 as well as it also lacks the NADP reductase enzyme. Because see I mentioned to you here that here only ATP will be formed. There is no NADPH formation. If there is no NADPH formation, then we do not need the enzymes also associated with NADPH. And that is why we can say that since cyclic phosphorylation is not going to produce NADPH, so it also doesn't need NADPH reductase enzyme, and that is why indirectly we can say that it lacks NADP reductase enzyme. It doesn't need only NADP for anything. Why will it require? So instead of saying it that way, our Ncert says that it lacks. So where it is written here only, while you will read, you will come to know. Here you can see stroma lamella membrane lack PS2, as I already mentioned, the first thing, and the second thing, NADP reductase enzyme. It lacks PS2, lacks NADP reductase enzyme. So this is how you have to take out the Ncert Ncert. Okay. Earlier there used to be one Ncert Ncert series. So basically this is how you need to extract the Ncert lines. Okay. This is the actual way of reading Ncert, which you will learn slowly, steadily, gradually. You will also learn one day. Okay. So do not lose hope. If you are thinking by seeing all these chapters, next day I will not take this, then do not do this mistake, because this is not that hard. Very interesting it is, if you see from my point of view. If you try to understand. And now on whatever topics, including this and whatever topics we'll be getting, in the upcoming pages. All are very, very important. All can fetch you 5 marks question if you come to the theory exams. Entrance exams also, anything might come. Entrance exam this chapter, and from entrance exam point of view, it's more important. And honestly, in biology, you cannot skip any chapter from NEET point of view. Every chapter will have its equal importance. Just some chapter might 2 or 3 questions might come extra, but all the chapters questions will come. So you cannot ignore any chapter. You cannot say that this chapter is hard, so I'm leaving it. Even in entrance, not too many questions will come, but remember, even if from here three questions also if it comes, now too I think more questions will come in the coming time, since lots of chapters are deleted, so automatically its weightage will increase. And 1 mark, there used to be thousands of rank inflation, thousands of rank difference used to be there. So you cannot ignore any chapter of biology. You must be excellent in all the chapters of biology, all the topics. You have to, take out the extracts of this chapter the way I'm taking out. Or for all the chapters, you have to do this way. If you study this way, you are in the right direction. Okay. So now let's come back to our topic. So I was telling you about stroma lamella, very important thing. In grana, you can just write it down in your Ncert also, that in grana just write a dash and write here non-cyclic phosphorylation will take place, Z scheme. It possess both PS1 and PS2 product, ATP and NADP. This was all with respect to grana I mentioned. Now with respect to stroma lamella, what you will write? You will write it is cyclic phosphorylation, cyclic pattern, lacks PS2, lacks NADP reductase enzyme. This is a point wise here only. Write it down for stroma lamella, for grana. So whenever you will be revising, you will see and you will recollect. And honestly, I will tell you, if all that while explaining whatever I have mentioned, that has helped you in understanding, if possible, write it down in the corner. This section is there for that only, to write down if you find anything important, anything extra you didn't understand earlier the statement, via my explanation you understood, write it down in your own language. Next time it will become easier for you while reviewing this chapter. Okay, so let's come back. So where were we? So where the cyclic, now we are in the cyclic, we are talking about this. So whenever we are in a cyclic photophosphorylation, it will take place in the stroma lamella, and non-cyclic will take place in the grana. You can write it down here somewhere, as I have mentioned. Okay, while the membrane or lamella of the grana has both PS1 and PS2. Stroma lamella lacks PS2, lacks NADP reductase enzyme, as I have already mentioned. Now, the excited electron does not pass on to NADP+, but is cycled back to the PS1 complex through the electron transport chain. NADP+, there is no mentioning. Okay, that is why it is saying that it will not pass to NADP+. And that is why NADP reductase enzyme also not needed. That is why lacks NADP reductase enzyme. This is how you will take out the meaning of the statement from your side. They will not directly tell you, but indirectly they have told you. You will have to understand it. Okay. So where I was? Yeah. PS1 complex to electron transport chain. Okay. The cyclic flow hence results only in the synthesis of ATP. So in cyclic phosphorylation, there will be only synthesis of ATP. Only synthesis of ATP. There is no mentioning of NADP, but not of NADPH plus H plus. Cyclic Photophosphorylation also occurs when only light of wavelength beyond 680 nanometer are available for excitation. Why did they mention beyond 680? Ponder upon it. Because here only PS1 is included and PS1 needs 700 nanometer, right? 680 was for PS2. Since here there is no mentioning of PS2, so no mentioning of 680, so it has to be more than 680, then only it will be 700. Yes or no? More than 680 is equal to 700, not is equal to, whatever. That is why it is said beyond 680 nanometer means 700 nanometer means PS1 means cyclic photophosphorylation. Understood? So this is all. Once again I'll summarize how much I can regarding cyclic and non-cyclic. How will you differentiate? Firstly, the location. Non-cyclic will take place in the grana, cyclic will take place in the stroma labella. In non-cyclic, second point of differentiation is PS1, PS2, it includes both. But in cyclic, it includes only PS1, 700 nanometer. Okay. Third point of differentiation is the product. Non-cyclic photophosphorylation products are ATP and NADPH. But cyclic photophosphorylation product is only and only ATP. And cyclic phosphorylation takes place just to increase the efficiency of photosynthesis. Okay, in a way helping the non-cyclic phosphorylation with the extra ATP required for the photosynthesis. So this is what. So I end up this here, and the next part, chemiosmotic hypothesis, very very important. People generally find it hard also. Till here okay, understandable. But here when you come to this diagram, seeing that only half of the people go mad. For them this diagram only becomes a nightmare. So don't take this diagram that way. If I just shared a tip that this diagram you need just for understanding the theory that is mentioned here. Nobody will ask you the detail of this diagram. And what is this plastoquinone, plastocyanine? These are the electron carriers. Just like earlier, in the earlier lecture, we mentioned about cytochromes. Electron transport system. Do you remember? Electron transport system consisting of cytochromes. Here they did not show cytochrome, but I mentioned in that video that cytochrome is an electron carrier. So just like that only, here electron carrier, just like that only, plastocyanine, plastoquinone, these are also electron carriers. They will carry the electron. If they get from, suppose from PS2 they got the electron, so they will carry that electron to plastoquinone. Then plastoquinone, it will go on, and then from plastocyanine will receive the electron again, it will pass it. Electron carrier because it is carrying the electron from one place to other place. Okay. This one is the thylakoid membrane, because I said thylakoid membrane inner space is lumen, outer space is stroma. Stroma is the inner space of the chloroplast. Okay, inside the chloroplast there is thylakoid. Okay, if you think this boundary of the book is suppose the chloroplast membrane, then inside the chloroplast there will be thylakoid. Inside the thylakoid there is lumen. So this inside portion is lumen. This is the thylakoid membrane, and outside the thylakoid membrane is the stroma. So here we will talk about the gradient, okay, about the potential gradient across the thylakoid membrane. Chemiosmotic hypothesis is all about that. We'll cover in the next chapter. If you have any queries, you can ask me in the comment section. And this is easy, you will understand. Okay, so thank you.